All right, so let's start a very short portion and we will go quickly over that. Quickly means like whatever time it takes. And after that, we can just kind of go over the material in 3.1 and 3.2. Just an overview of that. You already have the test review. So rather than going through the test review, I just can pull up some of these PowerPoint slides and just talk about some of the areas that you need to focus. And again, it is completely optional. So it will be recorded as well. So if you need to leave for any reason, you are most welcome to. There is no problem with that. So completely up to you. All right, so let's start with the module 3.2 part 4. And we are talking about really an extension of the stem cells, and mainly, although it is still written as a stem cells in there, this is really, we are going to talk about the cloning. So if you remember from the previous section, or the previous part 3, we talked about two of the scientists, and we talked about Dr. Gordon, and he actually pioneered this somatic cell nuclear transfer. And that's what we are going to talk about today. And so that we just have an overview of the material. So here we can easily see that the somatic nuclear cell transfer, what we mean by this. So just to let you know that you all know that this is the famous sheep dolly, which was the first cloned mammal. And let's take a look at how we do this and what is this process and how it is different from the stem cells. Now, the stem cells, we already talked about that we have two different kind of stem cells that we have used. That one is the embryonic origin, embryonic stem cells that you do obtain from the inner cell mass of your blastocyst. Say, for example, right here is a blastocyst and right here is the inner cell mass. You just get those from there. Or you can harvest them from adult stem cells. So we talked about that, where these stem cells are, say, for example, intestinal crypts and other areas, and you can reprogram them. So these are some of the things that you can use. So let's take a look that, just 1 second. Okay, yeah, everything good. So let's see that what we do in the somatic cell nuclear transfer, which is very different from the usage of your embryonic or the adult stem cells, which is entirely of a therapeutic purpose or some other purpose. So here, what you are seeing that when normal development of any of these mammals actually do happen, including many of the lower ones, you have an oocyte, and right here you have this, the chromosomes are 23. Remember that we have 46 chromosomes. 46 means we have 23 pairs of chromosome. What it means, 23 pairs, that you have chromosome number 1, you have 2 of the chromosome number 1, one from your mom, one from your dad. Then you have 2 of the chromosome 2, one from your mom, one from your dad. Whereas here in the oocyte, before it is fertilized, you have just only one set of chromosomes. So you have only one chromosome, that is your mom origin. You have only one chromosome of chromosome 2, and the same way, you have 23 of chromosomes, and that's why we call this stage as 1N. And if you remember from your Bio 101 or genetics, that we call this condition as a haploid. Haploid means you have a single set of chromosome. So the same way, the sperm from the dad origin is also the same way. You have chromosome number 1, chromosome number 2, chromosome number 3. All of them just have one copy of that. That's also the haploid. And once the fertilization happens, you form the zygote, and now it becomes diploid. You are seeing that still there are 2 nucleus. It didn't fuse yet, but they will fuse both of the nucleus, one from the mom, one from the dad, and it will form the 2N zygote, which is the diploid. And then all of these cells will undergo this condition, which is the blastocyst, and that implants into the uterus, and it will develop into an adult within the uterus of a mammal. That's the normal way. Now what you do, that in a reproductive cloning, what is done, that you isolate the eggs from the mammal. And this can be done even in human beings to treat fertility. We call this in vitro fertilization as well, but we'll come to that later. This is not exactly the same. In vitro fertilization to have the babies the people who are just suffering from infertility, that's an entirely different thing. That's not reproductive cloning. So anyway, the idea is that you get the mammalian oocyte or the egg. Oocyte is just like a scientific term. and then you enucleate the egg. And how you do that you take a microscope and you will hold this embryo with a glass pipette, and you will just use a very fine glass pipette to puncture through the cell, and you can suck this out. And these are used by some of the sophisticated microscopes attached with something which is called a micromanipulator. And while you are looking at the cell, you can do that. And when you enucleate the egg and take out that glass needle out of the egg, it's the plasma membrane seals back, and there is no problem, no leakage of the material. So once you enucleate the egg, also along with that, you need an adult cell. Say, for example, any random mouse that you need to clone. You get an adult cell, and usually it could be a skin cell biopsy. You could get a fresh skin cells, dividing cells, fibroblast, any kind of cells, which all of them will contain the diploid number of chromosomes. Now, you take the nucleus the same way you enucleated and took the nucleus out here, which did have the 1N, but here you have the 2N, and you transfer the nucleus back into this enucleated egg where you have the 2N. Now what will happen, as you put the nucleus in here, you can actually in vitro just put some of the growth factors, and this egg will differentiate into the blastocyst, and you can put this one into usually a surrogate mother. You can put that in the surrogate mother, and now, fortunately, that will develop, and that's been developed many times, although it is an extremely difficult technique, but it can be done. So it will develop into an animal. So the advantage is that what is happening, suppose this is your adult mouse. If you take a cell from this adult mouse and put that into an egg, the developed mouse inside this surrogate mother will be an exact replica of this mouse. Why it is so? Because see that here, whenever a natural reproduction occurs, you get half of your mom's chromosome, half of your dad's chromosome, they mix together, and you have a mixed genotype and phenotype as well. Like you have a mixed gene pool. Whereas here, there is no fertilization. You are taking the whole set of chromosomes through the nucleus of this adult mouse, and you're putting in the egg, so it will be an exact copy of that. And in that sense, think about that when you are cloning by the somatic cell nuclear transfer, why we now say that, somatic means the cells of your body. So you have two types of cell. You have a somatic cell, and then you have a germline cell. Yeah, I will come to that. So somatic cell is any cell from your body, whereas the germline cells will be only the sperms and the eggs, which are 1N. So we have a questions that can it be any type of donor cell from the donor mouse, or it does have to be a bone marrow cell? No, I mean, like usually most of the time, it is not a bone marrow cell. Most of the time, see that getting a bone marrow is an extremely painful process. I mean, like you have to puncture the bone with some really difficult techniques. So when you do undergo these kind of therapies where you need the bone marrow transplantation, it's a very painful technique. All you have to do is just take a skin biopsy, just a small piece of skin, which will not even hurt you, and you just get a skin cell keratinocyte from very base germ layer, which is completely healthy. You can get a fibroblast. It could be any kind of cell. You do not have to get any. So to get this right here, adult cell, you do not have to get any stem cells. This is a general cell from your body, not an embryonic cell or stem cell. So the next question is that how can the differentiated cells form all the tissues? So that's the thing. So for the differentiation, the factors that are in there is all in the egg. So what is happening right here? You are transferring the nucleus to the egg, and all the egg that is basically understanding is that once I have a diploid nucleus, I need to differentiate into the blastocyst. Here it is occurring naturally. You're getting the sperm and the egg nucleus. When fusion occurs, it just undergoes into the blastocyst formation. Here, as you are transferring a whole nucleus in the 2N condition, it will undergo into this nuclear transfer into the blastocyst. But it is not as that simple. This is a good question. There are some external growth factors that is needed, because think about that. This differentiation is happening usually in the uterine tubes or the fallopian tubes, and there are some external stimulus that's in there. There are some growth factors that is needed for this egg to develop into the blastocyst. And when you do this in vitro fertilization, you provide those growth factors outside and it will differentiate. But most of the triggering factor is already in the egg. All it does is to understand that if I have a diploid nucleus, I will be able to differentiate. Does that make sense? Good?
Yes, Professor.
Okay, great. So now it will develop into that. So that's the overall process of the reproductive cloning. So the next thing that what we are coming into, that this is a kind of a funny picture, that over 20 different animals have been cloned. So you can see that. But there are a couple of the different things that I want to point out. So this one, I think that it is a great name and just made the news, which is the copycat, or CC. I think a very, very clever name. So that's the copycat, and you can see that here is what you think, is she the mother? But this cat is a nuclear donor. Now here, there is a very important thing that you can see that what is happening. So as I said, that when you do the somatic nuclear cell transfer, you should get an exact replica of this cat to this cat. Now, what you see, that this is the nuclear donor, but not the mother, you should think that this cat is a twin of this cat. The only difference is that they were born in different time scale. This twin one born earlier, you get the somatic cell nucleus from there, and you develop the next one. But what is most important here is that see the difference in the coat color. The coat color of this kitten is entirely different from the other cat. So what it tells you that the coat color is what is called a polygenic inheritance. That means that there are many, many genes responsible for that. And these genes are controlled by the environmental factor where they are growing. So it depends upon the different factors of the surrogate mother where this embryo was incubated. So that's a huge difference, that although they are genetically the same, remember that, genetically the same, they are different. And you can just easily tell that, again, we have identical twins, like naturally born identical twins. And as they develop, as they grow, they will have a little bit of different characters. They are not exactly the same. And some of these things are influenced by the environment. So that's one of the important things. Many animals have been cloned over the period of time. I think that the latest one was an Arctic fox as well, because their numbers were declining. So there are been many of these things, but this is just for your informational purpose. So the interesting thing, what happens, that when we cloned the primates, the monkeys, at that time, all these ethical issues arose, because up to this point, okay, all right, it was a scientific fun, and you can just argue that, okay, all right, we are just making livestocks. That could be really, really great. But once it came to the primates, then the question is that then probably cloning humans is also possible, and we'll come to that. On the other hand, this is a really ambitious project that is going on, and it's still there. A group in Harvard, along with several, this is a worldwide collaboration. Many different labs are working on this, and there is not a success yet. So what they did, that these scientists extracted the DNA from the frozen specimen, permafrost of the tundras, and these woolly mammoths were extinct 4,500 years. But DNA is extremely stable as such. It's not unstable like RNA. And you can get good amount of DNA from this frozen tundra. It's not completely probably intact, but you get a good amount of DNA from there. Of course, you cannot get an intact nucleus. Then it would have been not a problem. So now what they're trying to do, that they are taking a backbone DNA of the genome of an Asian elephant. And using this CRISPR-Cas9 technology that that you can edit the gene, they are trying to replace the homologous genes with this DNAs of this woolly mammoth. And what I mean by the homologous gene, think about that if there are some of the specific genes that are required for this different type of tasks, and compared to a smaller task of the Asian elephant, they are replacing that with the woolly mammoth gene. Suppose, on the other hand, if we just go to the molecular level, say, for an example, the cellular receptor genes, or say, simple as a sodium potassium pump, I'm just saying, just randomly, these may have a little bit of difference in the woolly mammoth compared to those. And they are doing this homologous recombination, or which is based by this CRISPR-Cas9, to edit the gene which is based on the backbone of an Asian elephant, to replace them with a woolly mammoth. And it's an enormous, huge task. I mean, like, think about that. It is a 3.2 base. Human genome is about 3.2 billion base pairs. So it is about 3 to 4 billion base pairs that at least some part of it needed to be replaced, and it is not an easy task. Many different laboratories, multiple years, big groups, but what they could do, that after they could manipulate the DNA, they could put a cell, basically the Asian elephant cell, and they removed that nuclei, which is the oocyte, from there, and then they used that oocyte the same way that they packed all of these in a particular cell, and then these manipulated cell nuclei is put in there, and then the eggs stimulated to turn into embryos. So this was one of the questions, that how can a differentiated cell from the tissues? And here you can see that the eggs is stimulated, as I said, and that should form the embryo. So this is yet to be developed. So they got these embryos, but they somehow, and see that this is the disadvantage, is that, so where the fine balance is, you can get all the growth factors to turn this embryo into a blastocyst. But probably what is happening, that there are some growth factors which are from the woolly mammoth, and the scientists are still not sure, or did not find the proper balance, that to grow this with this replaced woolly mammoth genome into a blastocyst, you need something that has not yet been discovered. So it is not really an easy task. So it's still going on. So that's an overview of that, where the cloning process stands. So here is a kind of a funny story, although I just put big pictures of that. I should put pictures of Nobel laureate that big compared to that. So there was a company, Clonaid. Here I'm just going to just go a little bit. So this is a Canadian company, and they registered in the Bahamas, not in the United States, not in a club. And probably this is just to bypass all these rigorous laws that is in there. And Dr. Boislier, she is a French Canadian scientist, and Dr. Antinori is from Italy, and he is a doctor, MD, and he treats infertility doctor. And they claimed that they have cloned the first human baby. And if you just look with this name and Google image search, you will see that there are pictures still in the Google with Dr. Antinori holding babies and saying that he has cloned several babies. Unfortunately, both of them, this is a hoax. Both of them were arrested, and I think that he's spending his time in jail, unfortunately. So yeah, people will do anything to do, come to the fame. I mean, like, they were all over the news channels and everything. And this was done by another scientist in Korea, and he retracted his claims as well, which turned out to be false. So far, there is no cloned human beings as such. There is no evidence of any cloned human beings. So purpose, this is very general. Agriculture, champion livestock, animals with favorable phenotype, absolutely acceptable. That is fine. Animal model for the disease, this is also another interesting thing. So this is very perfectly fine that you completely understand without going into details, a particular type of meat, a particular type of growth, development, those are all great. On the other hand, what happens, this is an interesting thing, that why you need animal models for disease. So think about that when you are testing anything nowadays with this COVID-19, the people are trying to develop the vaccine as fast as they can. And the first thing that they do in the preclinical trial is using, say, a mouse model. Then they may use a ferret model. Then they will go to a primate like a monkey model. Then they will use a human trial. So they will use these animals, and these animals, individual animals, are not exactly the clone of each other. So each of these animals will have slightly different physiological and immunological parameters. Although they can be born from the same mom, but remember that they can be born from the same parents, but still even that your siblings exactly don't look like you. All of them are a little bit different. The reason that during the meiosis that happens prior to the germ cell division, at that time you have the crossing over, you have the mixing of the parental chromosomes, and that mixing is just random. It is not exactly the same. So even if the offsprings are born from the same parents, they will have a slightly different physiological parameters, genotypic as well as phenotypic. So what happens that when you do these animal models for this disease, you will get a kind of a mixed result. So due to that, what you have to do a large population so that you normalize the discrepancies in between the individuals. Say, for example, you may have to do like 10 sets of 30 animals together, because there is some differentiation and variation in between them. And then you can normalize and make a standard deviation to tell that, okay, all right, this part of the data is outliers. So this is conclusive, this is not. So it makes it much more expensive. On the other hand, if you just do a clone, then you have suppose these mice, they are exactly the identical of each other. Although you can argue that they may have environmental effects, but if these are basically born in the same laboratory conditions or animal facilities, they are exposed to the exact same environment, and chances of them to be genetically and phenotypically close or almost identical is much more than the naturally born. And that will reduce the cost of research. So that's one of the great things that the scientists are arguing, that why cloning is important for them. That's great. On the other hand, the last one is the human cloning, which is the reproductive cloning, live birth cloning. And this is absolutely controversial, and I'm not sure that why somebody will like to clone them. It's kind of a weird thing, I guess, but by all ethical purposes. On the other hand, you can do a therapeutic cloning. We will come to that. And this does have the potential, the same way that we are doing the stem cell research. Adult stem cell research has been used for the therapeutic purpose. And this is also just a little bit ahead of that stem cell research. And we're going to talk about this, that what therapeutic cloning is. So before going into that, this is just an overview of that as I said, that as an infertility treatment, a bad idea. Well, you can just do that to the farm animals or laboratory animals, but live birth cloning for the human beings is not a good idea. And also it is extremely inefficient. You do not have to remember any of these numbers, but before Dolly, 277 attempts and 29 made it to the blastocyst stage. So you can easily see that how huge number of these cloned embryos you have to do that. And that's what it is. So one thing I just forgot, that why this Dr. Antinori was in jail is not due to the fake news that he put, but actually there was a lawsuit filed by one of his patient, her patient, both of them, and the patient actually basically complained that the doctor gave him some of the drugs and tried to remove her oocyte without her consent, and that's absolutely a jailable offense, I guess. So that's what they were trying to do. Anyway, so that's the overview of that, the bad idea. On the other hand, the second thought is that cloned animal may not be normal. So Dolly was cloned from a 6-year-old sheep, and it has early onset of arthritis and cancer, as if the Dolly was not at his age and already 6 years old when it was born. The Dolly was born at that time. On the other hand, that the scientists also have seen that cloned mice have shortened lifespans as well. But there is good amount of research now that is taking place that they have seen that this may not be the case. They have overcome these issues that the cloned animal they are not normal. Their gene pool is not normal. There are some problems from the epigenesis, which are outside factors of their genes that they are in there. So now you can see that the cloned animals, there are plenty of evidences that cloned animals do have the same vigor as a normal animal. So that has been overcome. So this is what is important, that is we are trying to talk about, and that is the advantage of your somatic cell nuclear transfer cloning or the therapeutic cloning. So what you do here. So here you have a patient, and you will take a cell from the patient, and see that right here, somebody asked, there's a somatic cell biopsy. So you really do not have to take any cell from your bone marrow. I mean, like, if you wish, you can, of course, but why you would do that when you can take a skin tag literally without any harm and just putting a Band-Aid on top of that? You can get some somatic cells, very healthy somatic cells. For example, skin cells. And then what you do, that you take an egg, and you have to have an egg donor. Remember that you have to have the egg donor. And you can always question that if it is a female patient, can you use that female patient's own egg? Of course you can do that. So you have the choice. You can use the same egg from the donor if you have the ability to do that, or you can get from another donor. So then what you do, that you just same thing, enucleate the egg, you transfer the nucleus into that, and in vitro, this is all happening in the Petri plates. You add the different growth factors, and this will differentiate into the blastocyst. So this is basically an embryo that is formed from this is a clone of your own set of genes. That's basically what it is. So once this blastocyst is formed, you take the blastocyst, you have to destroy the blastocyst, and you take the inner cell mass. You remember that the inner cell mass are the pluripotent cells, which can develop into any type of tissue. So now you take these embryonic stem cells, these inner cell mass in the culture, then you can differentiate them. And how do you differentiate that? For each of the different type of tissues, it has already been identified that there is a specific amount of growth factors that is required. This is extensive research going on, but the scientists have successfully developed into pancreatic cell, hematopoietic cells, say for example, the bone marrow cells, cardiomyocytes. So here, neurons, hepatocytes. So these are all the possibilities. And then what you are going to do, that you are going to take these cells, which are still growing into the Petri plate, and you put that into that individual. So here's a huge advantage of that. So this is immunologically compatible transplant, because what is going to happen, that once you having these cells going into your body and you differentiate them into the hepatocytes, there will not be a tissue rejection. They can take over, because the genotype of these cells are exactly the same. So this is the advantage of the therapeutic cloning. So let's take a look, a little bit of detail, which is not required for your exam, and just to give an idea that why you want the hematopoietic stem cells to be delivered. So think about that. Here there is a problem. Say, for example, a real life problem. Suppose a patient do have a bone marrow cancer, some kind of leukemic cancer, where the bone marrow is dividing into the cells uncontrollably, which is basically blood cancer or bone marrow cancer, some site of leukemia. So at that time, how you can save that patient? And this is reality. This is this is being done already. So you take the patient and you will irradiate the bone. That's a very hard part. You will irradiate all the bones and kill all the bone marrow cells. That patient will not have any bone marrow cells that is alive. So multiple time radiation, along with that, many of the blood cells will also be killed. So again, you can transfuse blood and make the patient okay for some time, but your target is basically to kill all the bone marrow cells. Advantage is that the bone marrow in in an infant or a young is all over the body that is producing the blood cells, whereas you have some specific areas of the bone that does have the hematopoietic cells. All bone marrow everywhere doesn't have the hematopoietic stem cells. So you can just preferentially do that. Then what you do, that you differentiate these embryonic stem cells to hematopoietic stem cells, and now you inject these hematopoietic cells back into the bone marrow. So now you have a healthy set of these hematopoietic cells that is going inside your body and will reproduce normally. Now, I skipped a big part right here. So remember that at the very beginning that I said that if you have a specific gene that is responsible for your bone marrow cancer or a blood cancer, you take this somatic cell, you edit that gene by the CRISPR-Cas9 so that you have a normal gene now. So you repaired the gene, then you take that nucleus and put it back. And that will give you a normal hematopoietic stem cells, which was not there in the patient. So this is the power of that. And the same thing, the cardiomyocyte or the neurons in the neurodegenerative disease, if a person lose a part of the kidney due to some, need to do a kidney, I'm sorry, not the kidney, the liver transplant. If a person needs liver transplant, you can save the liver by transplanting these embryonic stem cells. So this is the main goal of the therapeutic cloning, and it is already in the process. Any questions? Wouldn't the shortened telomere from the donor will be an issue? Yeah, so that is a very good question. That's always there. There's always, we will age, and no matter what, this is of course an issue. So that one of the questions that has been asked, that when you are taking the somatic cell that is from the adult, so as we know that as we age, we constantly lose these telomeres over the period of time. Now, the thing is that, remember that, this is a very good question, that you have to take, and that's one of the skin cells that what we do, that when the skin cells actually divide, a single skin cell will not divide more than 50 times. So the chances of those are that the telomere shortening is of course a problem, and that will give a premature aging of the cloned animal. But we try to minimize those, because depending upon that from where you are trying to get your cells, some of them will grow much faster. So most of the cells in your body will not divide more than 100 times. So that's usually the therapeutic limit or in there. And what you can do, that when you actually do isolate the cell, you can tell that from the stem cells how many times that cell has been divided, and you have these telomeric markers that you can do. So there are some intricacies that you can do, and the scientists have found those keys that you can tell the telomeric length that, or the age of that particular cell. From there you are taking the somatic cell, and that's one of the advantage of that. So these are some of the things that you can overcome, but there is definitely, I'm not completely familiar with a particular literature that may point out to those particular issues, but there will be always those issues. It's always not 100% that we can just guarantee that it will be exactly the same. All right. So now the last part is that this is for your food for thought. What is the moral status of a human embryo? And these questions we are not going to ask in the questions, but there are plenty of bioethical course that you can take in the college as well. And scientifically, the embryo is a human being, but is the embryo a person or a piece of property? So there is a current surplus of frozen embryos, that there are banks of embryos that can be used. And the question is that who owns these embryos? Is the patient or who it is? So these are some of the problem. On the other hand, the last thing is that you can do these experiments to scientific freedom for scientific stewardship, but does that mean that you can do anything? So these are some of the questions that is out there. You can think there are plenty of books and literature out there discussing these things. So we'll just probably stop there. All right, so let's do the quick quiz. So the first clicker question will be, in order to clone your pet cat, which of the following individual component will not be an absolute requirement? What I mean by the absolute requirement, that you may or may not use it. So an enucleated oocyte egg from your pet cat, from a different cat, use your pet cat as surrogate mother for the cloned embryo. If it is a female cat, take that. None of the above are absolute requirements. All of the above are absolute requirements. All right, stopping it now. Good, everyone? Okay. So mixed reaction everywhere, but the correct answer is the D, which is still 38% highest. None of the above are absolute requirements. Now, let me explain that how it is so. So here, the first one is an enucleated oocyte from your pet cat to clone that. So you do not need that. Remember that you can get an oocyte from any of the cat, and you can just transfer that. So right here is an enucleated oocyte from a different cat. As I said right here, that you can get a different cat, but if your cat is a female cat, you can get the oocyte from your cat as well. So both of them are optional. You can get an enucleated from your pet cat, or may not wish to do that. You can get it from a different cat, or you may not want to do that. So none of them are absolute. Use your pet cat as surrogate mother for the clone embryo. If it's a female cat, that is also absolutely not required. A surrogate can be any other cat, and not many people answered that. So none of the above are the absolute requirements, and of course all of the above are not correct. So can anybody tell that which one thing will be an absolute requirement in those that is not there? Can anybody talk? What will be an absolute requirement that is required from your cat?
Adult somatic cell.
Yeah, that is exactly the correct answer. You need the somatic cell, a somatic cell with the nuclei from your pet cat. That's the nucleus you require to put either to the enucleated oocyte of the same cat or a different cat, and then put in the same cat or a different cat as surrogate. So you need a somatic cell from your pet cat. That is the only requirement. Other than that, these are all flexible. Does that make sense? Make sure? Good, everyone?
Yes.
Okay, great. So let's go to the next one. And here, read the question carefully. It's so which of the following is not an advantage of autologous embryonic stem cell development and differentiation in vitro? They have very high pluripotency. They have differentiated into many tissue types using appropriate growth factors. It can be used for treating infertility. They can be used to treat degenerative or damaged tissues without being rejected by the individual's immune system. All of the above are advantageous. All right, stopping it now. All right, so 51% answer C, it can be used for treating infertility. That is not correct. See that why it is so. See that here it is written that not an advantage for the autologous embryonic stem cell development and differentiation in vitro. What I mean by this, that when you take these embryonic stem cells and differentiate in vitro, you are basically destroying the blastocyst, taking the inner cell mass and differentiating it. So that cannot be used for treating the fertility. You are not using that for treating the fertility. First of all, that's unethical, but unethical issue is not the thing, that you have taken those already out of the blastocyst and differentiating in vitro. And all of them have a very high pluripotency. That is correct. They can be differentiated into many tissue types using the appropriate growth factor. So you can just use, say, the nerve growth factor to differentiate them into neurons, or say hepatocyte growth factor to differentiate them in liver. That can be done. And they can be used to treat regenerative or damaged tissues without being rejected by the individual's immune system. So remember that this is an important one. Autologous means your own tissue, your own cells. So you take your own cells and then put that one in an enucleated egg, and then differentiate it to a blastocyst stage. You just grow that up to the blastocyst stage, and then you collect that stem cell from the inner cell mass and differentiate using the growth factors. And that will be the same. It will not be rejected by your individual immune system because you are using your own cells, and all your tissue histocompatibility complexes will be the same. So you have exact tissue match. Good, everyone?